right? From thousands of years, even every year now, there's like a festival, they call it Kumbh Mela, where millions of people go there.
But it's because it's so infected, right? Like he went there and he's like, people who have like whatever it was, like cholera or something, or diphtheria or something, and they go in there and they cures it. So like it's because there's so much of that toxin in the water, and so there's all these phages against these bacteria. So if you can pull them out, you can treat some of these diseases.
Have your own phage bank and then help create new classes of antibiotics.
What's cool is, so every phage has a protein. It's called a lysin, and that's sort of like the torpedo that the phage uses to break out of the cell. That's what a lot in the Soviet days during World War II, possibly even World War I, like there was a lot of work on from the Soviet side. A founder is just a historical statement. I founded a company, I'm a founder. CEO is a job. I have certain tasks I do for money, and entrepreneur is a mental disorder that makes you
crazy.
You have to do this and you can't stop.
What are your thoughts about aging? Do you think that's a disease? Welcome to this episode of Talk is Biotech. Today I'm going to speak with Ivan Liachko. He's the founder and CEO of Phase Genomics. We're going to learn everything about his entrepreneurial journey. How did he turn his lab research into a successful biotech startup in Seattle? Let's start with a quick introduction. It's already recorded.
You're already recording? Oh my God.
I don't declare it because, I think natural conversation is the best conversation. But if you start with your quick introduction, your story.
Sure. So my name is Ivan Liachko. I'm one of the founders of Phase Genomics. My backstory is when I was 11 years old, we immigrated to the U.S. from what was back then the Soviet Union. Now it's Ukraine. We lived in the Northeast, so I lived in Philly for a long time, lived in New York. And then I started doing science early on. So I'm a little bit of an oddball because a lot of people for their, when they're kids, they want to be like astronauts and stuff like that. And I just always wanted to do genetics. Like, I don't know, since I was a kid, my grandparents encouraged me and stuff like that. And so I started working in a lab, in a in a molecular biology lab when I was 16 years old. So I was still in high school. And that kind of was, I got this like summer fellowship sort of thing for high school students, and I started doing lab stuff, and then I just kind of like wouldn't leave them alone, and I just kept going back and like, give me another job.
Yeah.
So I worked in labs basically continuously since I was 16 years old. Through college, through grad school, all that stuff. And that's all I've really ever wanted to do is to be a scientist, to be a geneticist. And I moved, so then I did my PhD I did my bachelor's at Brandeis University in the Boston area, in Waltham, Massachusetts. And then I moved to Seattle. Then I went to Cornell for my PhD, Cornell in upstate New York. And then I moved to Seattle for my postdoc. Did my postdoc here at the Department of Genome Sciences. And then, I was kind of like a lifelong scientist at this point, right? Like at this point, I was doing science for like 20 years or something.
Right.
Because you're like high school, undergrad, grad school, postdoc. They were going to drag me out of the lab. Like, you know what I mean? Like, I would be holding the pipette, they'd be like pulling me out, my dead body out of the lab. So I was sort of, I was going to be sort of like, a lifetime scientist. And then we invented this technology, and then it was very impactful. It was very useful. We were doing collaborations with a number of labs, and it was very clear that this tech we came up with was solving a huge problem in the field, and it was doing things you couldn't do with anything else. And it was also clear that because it was kind of complicated, keeping it within the academic sphere means like nobody would like it would just not see the light of day. Like
that's fine.
There would be some labs, it would be like 3 labs in the world who figured out how to do it, and but it wouldn't go anywhere.
Right.
And I was like, look, we have to do something about this. We have to like turn this into a commercial product. Otherwise, it's going to be basically like a crime against science.
Yeah.
And, I convinced the leadership. They were very cooperative. They helped me spin the whole thing off. I sort of went up and I said, look, I want to spin this off as a company. I have no idea what I'm doing.
Yeah, I would love to double click on tech piece. How did you come up with it? But going back to your childhood or even your teenage years when you started working in the lab, like what was that experience that basically convinced you that's what you want to do for the rest of your life? Like when did you see the DNA first time, or what was that aha moment?
I don't know if I had a specific aha moment. I think throughout my childhood was just always very interested in it. I mean, I remember there was like this book I had when I was a kid, some like crazy encyclopedia. This was like the Soviet Union days.
Right.
So I don't even know what was in it, but like it had lots of like biological, like illustrations in DNA, and it just always seemed so interesting to me.
Right.
Right. And so there was family encouragement to kind of pursue it too.
Right.
And so that's what it was. So it wasn't like a one-time thing.
Right.
It wasn't like, I had like, there was no specific event. Like somebody died from a disease and I said, I have to cure this disease. Like it wasn't anything like that.
Right.
I just was super interested in the biology of it and how it all works.
Right.
The technology to mess with DNA and molecules and cells and stuff like that just always seemed very compelling. I always wanted to do it.
Right.
So, and it just kind of grew from there.
Do you remember the first time you saw the DNA, and like how did you extract it from something?
Yeah, I mean, maybe not the exact first time, but yeah, I mean, when I started in high school, when I was doing, I did my first sort of DNA extractions and the plasmid preps, these things that we do to extract little DNA circles, purification, and I ran my first like electrophoresis gels and stuff like that when I was still, like 16 years old. Like it seemed like super cool and like
I know.
It seemed like real science, it wasn't just like school science where you're like putting leaves under one microscope slides and stuff, but like real.
100%. I extracted DNA first time from strawberry using some detergent and all the household.
The glass rod and
I mean, I was like, it was very fascinating that this is like a blueprint that determines what should be the color, what should be the shape of anything. Right? It's really fascinating.
Right. It is crazy to think that. It's a sequence of letters,
right?
But it makes shapes, colors, right? It makes strawberry look like a strawberry,
right?
All this stuff, and it's all just information in this long molecule.
No, it's crazy.
It's kind of crazy, right? So,
yeah,
and that's kind of what the premise of our company, right? Phase Genomics.
Right.
So the general premise is that, like what you're saying, like it's information encoded, in these like that's what life is, right? Like life is basically information, just being transmitted, and how do you build these kind of biological structures? And so the field that I'm in, genomics, for those of you who don't know, listeners in the invisible world,
we'll find out.
My invisible audience. Genomics is basically genetics at large scale. So it's like genetics, but instead of looking at one particular gene at a time, you're looking at like all of them, right? You're looking at how your genome, ome means everything. So like all your genes, what are they doing at the same time? And it's really, genomics as a field is really about trying to extract as much information as we can out of, these biological systems, trying to understand like what is the genome telling us in like a more large scale way, not looking at sort of one gene at a time, but like all of them. What are they all doing? Because most biology is super complex, and it's easy to think like in the popular literature or something, like I found a gene that does this, but there is no, in most cases, gene that does this. Like like a gene is just one tiny piece of a much more complex process. And so genomics tries to understand that. And the more information you get, the more things you can do. Right. And so that really what Phase Genomics was all about. We had this special sequencing, genome sequencing technology that let us extract a new kind of information, and you could do things you just couldn't do before without it. So what we've done is we've essentially we have the special way of sequencing DNA samples that allow us to do all sorts of things. Assemble genomes better, discover new microbes from microbiome samples, to detect like chromosomal rearrangements in cancer samples, how your genome's getting screwed up. And so it's this kind of like tech that has many, sort of a hammer that hits many nails kind of a situation.
What it's called, high C, something like that?
Yeah, so the original technology was invented invented at the University of Massachusetts.
Okay.
They called it high C, and there's a reason for why. It was there's a whole history behind the technology,
Sure.
but basically it was a special way of doing sequencing a genome in a way that told you which DNA molecules were touching which other DNA molecules. So the purpose was to look at how your genome is folding in 3 dimensions. So if you've ever seen a picture in a textbook or something of a cell, the DNA inside of a cell is squished like a kind of like a 3-dimensional sort of spaghetti ball, right? It's a long linear molecule, but it's squished into a little ball. But actually, like anything, like how it's all folded up is very important for biology, right? So you have some pieces that are touching other pieces, and that maybe will turn a gene on or turn a gene off.
I see.
So there is a lot of information in this kind of non-coding part of genetics.
Junk DNA.
It's this kind of like, it's not even junk DNA, because it's really just about where things are in the nucleus and where they are next to other things has an effect on how they work.
So proximity is important.
Right, proximity is important. And so the principle is behind that method is you're essentially you're using a chemical that glues DNA molecules together. And if you take a cell and the DNA is all inside of it like a ball, if you treat it with this chemical, the sequences that are touching each other will get glued together, and then you can sequence those little junctions. You can count how often every part of the genome is touching every other part of the genome, and from that you can calculate the 3-dimensional model of the entire genome, right? So you can be like, these 2 things touch each other all the time. These 2 things never touch each other, and you have that for every combination of sequence of spots on the genome, and you can make you can reconstruct a 3-dimensional model of the genome. And so that technology, that's the high C technology. That's what it was used for. And we took roughly the same technology and just said, wait, this information can be used for other things. So that this is kind of like the point is that it's about information, right? It's about like you capturing some information and figuring out new things to do with it. And so what this method could also do, so if you can tell which part of the genome are close and which parts are far, what it does is it allows you to reconstruct the genome. So imagine you had like the blueprints for a house, right? And you shredded them into little pieces, and then you said, here's the house, right? It's a pile of pieces. Technically, yes, that is the house, but it's not quite the same, right? Because it's just little you don't know where anything's going, and that's what genome sequencing is like. So genome if you were trying to sequence a genome for whether it's a human or some kind of animal or some sort of plant, what you're essentially doing is you're going to shred that genome into tiny pieces and sequence the pieces. And then you have to figure out how they all fit together, but you don't know. And so it's kind of like this this idea of taking a blueprint, shredding it, and now you have a pile of shreds. It's not the same, right? But now what if, right? So imagine, originally you had this blueprint that's a big piece of paper. What if you took your pieces? What if you knew how far away every piece was from every other piece? Right? What if you had a separate piece of information that said this piece and this piece are really close, this piece and this piece are really far, and it had that for every combination of pieces. Now, if you have that information, you can arrange those pieces in such a way, like to say these are supposed to be close, these are supposed to be far, and you put them all together in a way that fits. You can reconstruct that entire blueprint now, right? It's like whether it's a blueprint or a jigsaw puzzle, right? Like trying to put a jigsaw puzzle together. What if you know the distance between every pair of pieces? So suddenly this technology allowed us to reconstruct genomes because it told you how far away everything is from everything else. So suddenly you can take that shred of genome sequences and actually assemble them into full chromosomes, into a full genome. And so we started the company basically doing that for people as a service.
And during your postdoc, when did you realize it has commercial?
Yeah, during my postdoc is when we came up with. So I collaborated with there's a student in the lab. So this was a collaboration between the lab I was in, which was Maitreya Dunham's lab, and Jay Shanduri's lab. And there was a student in Jay's lab, whose name is Josh Burton, that came up with this idea of using Hi-C data for assembling genomes this way. We started collaborating, and then as we're doing that, then I had this idea that you can actually also use this to for microbiome discovery. So you can also we talked about this like jigsaw puzzle thing, right? Like reconstructing a jigsaw puzzle, and that's sort of what a genome is like. What it's like to sequence and assemble and build a genome for something like the Human Genome Project or things like that. But now let's say you're working with microbiome. So microbiome, right, is like, let's say what lives in your gut or what lives in the soil or in the water. You have thousands of different bacterial genomes. And you can't grow them in test tubes because they kind of depend on each other.
Right.
So you are sequencing them all together. So you take a bunch of soil, and there's a bunch of bacteria living in there, and you have to sequence all of them at the same time. It means you extract DNA from all of them.
Right.
And when you sequence that, it's like you're getting back to the jigsaw puzzle kind of that thing metaphor, except for now you have let's say 100 jigsaw puzzles, 1,000 jigsaw puzzles, and you dumped all the pieces together and mixed them up. Right now you don't even know which sequence, which pieces go to which puzzle.
Right.
You don't know which sequences belong to which genome because you have all these bacteria getting sequenced together. But if you know which pieces were touching each other, then you can tell which ones belong to the same organism. And so that was the basis of this sort of other facet of this technology, is that it allows you to take a super complicated microbial community where there's a bunch of different microbes living together, and you can map all the sequences that were touching each other inside the cells, and then you can basically separate out, you can reconstruct the genomes for all the different microbes that live in the sample. You can find the viruses, you can figure out which viruses are infecting which bacteria, you can discover where the antibiotic resistance genes are coming from. So it really basically just leveled up what you can do in the antibiotic, sort of in the microbiome space, in the infectious disease space. So that sort of has been the focus. And
it's really cool, right? So when we look at sequences, we think it's a very straightforward story, right? But there are many unknowable or invisible forces behind their expression, like how they're positioned, how they're structured. I wonder if gravity has a role if we try to express them in zero gravity, if that would make the difference too. This video is sponsored by SciSpot. SciSpot is on a mission to turn every biotech lab into an AI powerhouse. It provides the best data infrastructure to these labs.
Well, it does. We know that happens because people do this in space. I have friends who work for like space biology companies. But the proteins fold differently in low gravity. Genes express differently in low gravity. So people are doing a good amount of experiments.
I think if you culture bacteria in space, they will look different.
Well, I don't know if they'll look different, but probably will.
Yeah.
I don't know. I mean, you have different like pressure and stuff. That I don't know. But I imagine they will to some extent. But yeah, there's a lot of other forces we don't know about, right? Like gravity,
that's true.
And like light, and who knows what else, right? Like,
yeah, I mean, I find the whole space of zero gravity super fascinating, right? When you culture cells, they behave very different. And there are some companies trying to make space as a biomanufacturing hub.
Yep.
So I'm not sure in terms of genetic engineering or the whole genomic space, how it can be leveraged. Because gravity it adds another variable, right? If you remove gravity from the picture, it's one less variable, right? So you arguably have more clarity.
But the absence of gravity might also be a variable, right? I mean, I think we just try everything and see what works best.
Yeah, I see the difference.
Yeah.
Maybe growing plants there will be much more productive.
Because it's pretty easy to keep gravity constant, right? So it's not like a variable, and you can like have it or not have it, but you don't have to worry about like. So there was there's a story in the genetic space. So for a long time back in, I don't know when, the 90s or something, there was this problem. People used to do these things called microarrays, where they would put DNA onto these chips that lit up different genes. And a lot of times those didn't work, and people never knew why, because they couldn't figure out why. And then somebody eventually did this experiment, aggregating data from all these labs and all this stuff, and they figured out that it was like the success and failure rates were linked to like times of day and like seasons and stuff, and people couldn't figure out why. And then they realized it had to do with atmospheric ozone. And so basically ozone reacted with one of the key components in the microarrays, in microarray chemistry. And so if you did your experiment when there was a lot of ozone in the air, like on a hot summer day, it was going to fail. And if you did the same exact thing at night that day, it would be fine. And again, like, who would have thought atmospheric ozone is going to affect? So yeah, so gravity is a cool thing. All these other, but in that case, right, it fluctuates every day. Like ozone fluctuates every day. Gravity is the same. So at least gravity, you can kind of like
control it.
You can control it. You can be like, we'll do this. I'll have my lab here. Maybe I have my lab up in Mount Kilimanjaro.
Right.
but at least you have some control of that variable.
Makes sense.
Yeah.
Okay, so during your postdoc, you have some clarity on the tech. You understand the commercial viability. How do you turn that into a business?
So the way we started was we basically had people ask us to do this for them, and it's actually kind of a fun story. So there was an ag company who had a really good like genomics department, and I think it's mostly for research. I don't think they're like doing GMOs. I think a lot of it was just research and maybe assisted breeding and things like that. But it was the Driscoll's Berry Company.
What company?
Driscoll's. So if you guys have bought berries in the store, Driscoll's. And so they came and they wanted to do a bunch of genomes with us, and but they didn't want to, like, they didn't know how to use the technology, and so and it was all proprietary stuff, so we couldn't obviously publish it. And so they were like, look, well, can you like do this for money? And so they were one of our first customers. And I just went to conferences and we talked about it, and people said, hey, we'll pay you if you do this. And so we jump started it basically by doing like a service model, right? Where we started just doing it. My co-founder and I, we started doing it for folks ourselves, and then we grew the team. We were able to bootstrap the company that way, and then we grew the team. We packaged the protocol up into a kit, into a box. Scientists love kits, and then we wrote a lot of software to go with it, so people can start doing certain things themselves. And so we're kind of like this hybrid wet lab / software company. So we build applications around this technology, but we're also doing a ton of software development. We do our own, we do AI stuff. We're building our own. We're now getting to the point where we're actually constructing therapeutic molecules from a lot of this microbiome data we've collected, and so we're kind of doing a lot of things at the same time.
Right.
Yeah, but it all started by somebody wanting us to just do a thing for them, like
Makes
do a
sense.
favor.
So how do you? So, like, to start a company, you need many ingredients in the right mix, right? So you have idea about the tech, you have first few customers, then you have to figure out how to make this like a recurring business. Then you need to find your co-founder, register a company. So if you go deep, like, how do you start a company from university lab?
Yeah, I think first you have to have a team, right? Because you have to first say, okay, I have an idea and I have some sense for how I'm gonna like sell it and what to do with it. But once you have that, and you're like, look, I'm gonna try to turn this into a company, you need a team. Like, who's gonna actually do this work, right? And so finding, getting a team around you is really important. And so, and that comes from your network, right? You need somebody who will complement your skills, because you can't do everything. And so, for me, my co-founder, Sean, we knew each other socially for a long time, because I ran a D&D campaign in my house for a long time. And he was part of it. He was married to one of the other grad students in our department. And so we were, And so when he found out, and he was working at Microsoft at the time, he was a software engineer, and and so he found out, he's like, I want to do this. So we kind of partnered up, and I was sort of like the wet lab side of the equation. He was more of the software side of the equation. We split all the other tasks among ourselves. Accounting, contract, sales. I tended to be more like, he was the more like detail oriented person, and I was the more like kind of goofy person. And so like I would be the one traveling and selling and going to all the conferences. He would take care of more like contracting stuff,
operations stuff.
Yeah, and then and then we bring in more people. We bring in more scientists. We bring in operations people as the whole thing grew. So, but it's it's super important to really get a good mix of people around you, especially in the early days where you really have no idea what the heck is going on, right? What you're doing. So you really need, you need supportive leadership, because even the professors don't like actively do anything for the company.
Right, they're more advisory.
You need them, but you need them you need to get their blessing to do things, because the university listens to them. You need to, you need encouragement from them, just like as a human being. You need somebody in your corner, right? You need them to write you support letters for grant proposals and put their faces on slide decks and stuff like that. So you do need sort of this cloud of people around you. And even if you're kind of like the prime sort of pusher, you're the one who is just like going and doing all the stuff.
Right.
You can't do everything yourself, and so you have to have people you can depend on, and they have to complement your skills, right? Like if you're the person who loves to like do lab work and you hate being out on the road, then you need somebody who's out on the road and who's selling, right? If you understand how to do lab stuff but you don't understand how to do software stuff, you need somebody who knows how to do software stuff. And you just kind of you need to you need an effective team, right? You need an effective team. You also need, in the early days, because it's not the company is not really a company even yet. It's an idea. It's a project. A lot of people think of startups as like these get these images of like these high tech things with like all these shiny labs, and they're doing all this fancy science, but really it's more like a garage like full of crap. Like that's what it looks like. You're like got a bunch of stuff on eBay. Like when we started our lab at the incubator, it was just the two of us. It was literally like a large closet, and we dragged a bunch of desks in there from surplus, from UW surplus, where we carried these we got the desk for like $10 a piece. The desk was too low to be a lab bench, so I bought bricks at Home Depot and I put them under the desk. So we had like a lab on bricks. I outfitted the whole lab for like $10,000 of just like our own money. It was all eBay stuff, but that's when we started sort of that's how you started the ball rolling. And so it's not like an organized entity. You're just kind of like faking it till you make it. You're like doing what you can to like try to push it forward, and you're learning along the way. And so you need people not only who complement your skills, but people who will work well with you, because like you need to, like when you're building a team, you need to be like, okay, if I get into a fight with this person, what's that going to be like? You have to be able to like disagree. You have to be able to work together. You have to be able to not drive each other insane. Or if you do drive each other insane, like be the kind that you can like go apart for a while, forgive each other, and come back. You know, like that kind of thing. So really, it's that, and of course you need more experienced folks to help guide you and reassure you and all that stuff. Other entrepreneurs who have done it.
No, totally. That's why I started a business with my brother.
Yeah, because you've survived this long together.
I know.
It can't be that.
What can go wrong? We have seen all of that. So no, that's very helpful. So you need doers, but you also need people for credibility sometimes. Before starting business, I never realized how important is the credibility part, right? Because you're new, and people don't like to be guinea pigs, right? They want to make sure there's someone else who approves it, or someone who has worked with you.
You need someone to start the process, though, right?
That's
you
true.
You need to get someone to buy into you,
To bet on you in a
to
way.
bet on you in a way early on before you have credibility.
Yes.
And so you have to find some way to get that credibility started.
Yes. I think
Once you have one, they tell their friends, or whatever. So yeah. But absolutely, and like, how do you do it? Like, how do you go from zero credibility to some credibility?
Right.
You have to like talk to someone and convince them to take a risk on you.
Right.
All right. And to get that ball rolling. And so that's also really important that someone on your team knows how to do that.
Yeah. Even in early days when you speak with VCs or people are like, yeah, that's cute. But like, show me how will you make an impact? Share the success story.
Yeah.
So I think it is like initial, like a threshold that you have to cross. It does take some effort.
Right. Well, and in a way, they're testing you because it's a test.
It is.
Right? It's like, can they do this?
Right.
Because they're trying to measure your go-getterness and like how scrappy you are, and can you actually do it, right? And so you kind of have to get ready for that search for credibility, right? For that, like, how do I convince people that I'm legit?
Right.
And there's a lot of that.
And it's like a snowball effect, right? For us 4 years ago, when we acquired a few customers, then it became very natural, very like easier relatively to replicate that success, right? But what I realize is startups in early days really struggle with that initial fight for credibility.
I think some of that in our space comes from the fact that when you're a scientist in an academic setting, you think differently than as when you're kind of running a company.
That's true.
You think about you're really incentivized to like invent something or do something one time and then go on and do something else, right? Whereas in you don't need people to buy your stuff.
That's true.
Right? It's almost feels like, hey, I made this cool technology. If you don't like it, that's your problem. Like, that's like that, right? Like, you write a paper, there's a bunch of reviewers who say this sucks, this is terrible, and you're like, F you reviewer number one, F you reviewer number two, like, and so usually reviewer number one's the good one. So you're like, I love you, reviewer number one, reviewer number two, you're cool, reviewer number three, F you, I don't care about what you say. But you can't do that when you're selling, right? You have to think about what other people want more than what you want. And so and that implies you have to do things like marketing and sales. And when I was starting out, for the first several years, like, sales and marketing, to me, like, I was like allergic to it. Like I was like, I couldn't do it.
Most scientists are.
It just feels like it feels wrong. You're like, if this is good, then it should be obvious. People should just get it, right? Like, they should buy it. Why do I have to like sell and like tell people about it and like do like ads and Twitter and all this kind of stuff? But you get over that. But I think that's part of why it's hard in the beginning to find customers. Just the when you're coming out of academia and you're starting a company, just the concept of having like a customer is slightly offensive. You're like, customer. I don't even like to say that word, right? And so that there's a challenge having to get over that. I still, to this day, I feel like, sometimes I'll say like client instead of, because it seems a little classier than a customer. But yeah, so that's a thing to get over.
No, totally. And what I realize is scientists are more like artists, right? Best artists are not necessarily best business people.
That's right. And you're sort of selecting for kind of this lone wolf mentality. People who will just kind of like engage their, creativity and things like that to create something new. But those people can also be prima donnas, right? They can be they can be full of themselves. There's all sorts of like that stuff. Whereas, in industry, it's a different thing. In industry, you're like, I gotta push product. Like, I gotta go. So there's less room for
innovation sometimes.
Self sort of. There's less room for like what you want to do. You have to think about what other people want.
Yeah, because I think the main difference between science or any innovative work is that you don't have to draw the line, you don't have to draw the boundary, right? You can tinker, and tinkering is fun.
Super
right?
fun.
But if you want to turn that into a business, you want to think how to scale this,
Yeah, you have to
how to
stop
standardize it, right? How can you establish more repeatable sales? And all of that requires a trade-off.
Mm-hmm.
So it's very different worlds in a way.
It's hard, man. It's hard. We're terrible at knowing when to stop tinkering.
Yes.
We just tinker forever. Just trying to keep pushing the envelope. And it's really hard to settle on one thing, for us at least.
Yeah.
And to say this is the thing and we're just going to do that and everything else is out the window. Like, we're just focusing on this one thing. But focus is very important.
Right.
But it's very hard to do when you're in something like biotech, right?
Right. No, totally. So what is the application? You talk about ag science. What other applications are your core focus?
Oh, man. Again, so everything at Phase is really centered around the same technology.
Yeah.
The applications kind of, so there's what I mentioned about building genomes for plants, animals, stuff like that, microbes. There's a microbiome discovery aspect. There is an application in the cancer space. So when you, like, if you were to, like, let's say you got leukemia, right? You would go to, they would do these diagnostics on you where they look at your chromosomes under a microscope to see if your DNA rearranged. So chromosomes are, right, they're these things that in textbooks look like butterflies. But that's when your DNA sort of condenses into these kind of butterfly shapes. If your chromosomes rearrange, they can sometimes create new genes, like new fusion genes, and it can turn on and off particular, like, oncogenes or tumor suppressor genes, and it can lead to cancer for many different reasons. And traditionally, how people detect those things is by looking at the chromosomes and being like, okay, that, like, piece of a chromosome is a little bit longer than it's supposed to be. That it looks like that piece and that piece swapped places and stuff like that. It's a very, like, it's a super old school technique. Like, you're looking at DNA with your eyes, you know, like what are you seeing? Right. It's like 100 years old, but it's super entrenched. It's like everyone does this because a lot of this information there's no other way to get because regular sequencing has a hard time detecting rearrangements. Like, it's like back to that, like, jigsaw puzzle analogy. If you just take pieces and put them in different places, it's still the same puzzle. Just the pieces are in different places. And so sequencing has a hard time detecting structural rearrangements, right? So if you swap two pieces of DNA around, the sequence hasn't changed. You just put them in different places, and but it could be causing cancer. And so they do it with microscopes, and our technology can tell us when pieces of chromosomes are rearranging or scrambling or moving where they're not supposed to move. It's called structural variation, structural genomic variation. So that's one other aspect. So a lot of work we've been doing recently is really about detecting new chromosomal rearrangements, new gene fusions.
So patients is like a diagnostic business?
Well, it's not a diagnostic business yet, right? So right now everything we're doing is for research use only. It's for discovery. But yeah, and that's sort of the way to look at it in the future. So in the future, this would be, it's an assay that basically replaces a lot of these microscope-based assays with a sequencing version of those tests. So it's a much easier way to do with sequencing what you used to do with microscopes. So there are all these tests you had to do, and now you can do all of them in just one very quick library prep. And it also allows you to discover genomic rearrangements that you couldn't see because by microscopy because they were, like, too small or they were in a region that you can't tell visually what's going on, things like that. So it's basically an improvement over a lot of these oncology tests. So that's one thing. And then the other, that's our one of the major areas right now is basically, kind of looking at the chromosomal structure of genomes in cancer samples.
And you do this for biotech, clinical biotech? Or?
We do this with a lot of clinicians, so
Okay.
a lot of like folks at the University of Washington, at the Hutch. We have a number of labs that are starting to bring this technology in as tests, so they can offer it as a diagnostic test. And so we're working in that direction. We're working with pharmas and other companies who do, who are looking. There's lots of different things you can do with this tech. For example, if you're like cell therapy is very popular these days, right? Cell therapy is when you take cells from someone. So allogeneic stem cell therapy, for example.
Right.
You take stem cells from someone and you modify them, and then you put them back in to fight off cancer.
I see.
But when you modify them, you might be introducing cancerous mutations.
Yeah.
And so you don't want to put cancer into a person. And so you have to check those cells are clean.
So more like
before you
QC
put them in.
process.
Sorry?
It's more like quality check. QC
Yeah,
process.
like a QC for cell therapies. That's like one angle you can do with it. You can diagnose people with heme malignancies, with leukemia, lymphoma, myeloma, these kinds of things. You can discover new gene fusions and new rearrangements in different cancer types and solid tumor. We just published a paper on brain cancer where this head and neck, this squamous carcinoma, I think, where again we just discovered lots of rearrangements in these samples that you couldn't detect by other methods. So there is sort of a diagnostic element to it, which is you're like testing for the presence or absence of known things that cause cancer or don't cause cancer. And then there is a discovery element to it where you can now find new things in cancer samples that you couldn't before. But it's really all about it's like a better lens on detecting structural rearrangements in genomes. So that's one, and then so there's like the cancer side of things, and then the other one I mentioned, microbiome stuff. We've done microbiome stuff for years. We built one of the most advanced microbiome analysis sort of platforms, and we've collected a lot of data, and that data is full of crazy bacterial genomes, virus genomes, phage genomes, antibiotic resistance plasmids, all sorts of stuff. And so right now we have a big project underway doing basically the discovery of new antibiotics to fight off specific resistant pathogens. So bacteria that are resistant to normal antibiotics. We have the world's largest collection of these antibiotic proteins called lysins that kill specific bacteria, and so we're starting to take those and basically develop them into precision antimicrobials.
Love it.
So that's the idea. So there, it's like the cancer stuff. We're kind of building that now. We're building it to be a diagnostic, to be a test, etc. The microbiome technology, we originally launched it years ago, and we've been doing it for customers. But we've also aggregated so much data that now we're pivoting towards. Not pivoting so much, we're not going away from the original, but we're expanding into saying, okay, we have all this, we have this massive data atlas. Let's start making therapeutics with the data we've collected. So that's sort of that's kind of where it is.
So you think the future of the company is therapeutics, own products?
We think there's two. We think, to be honest, that the oncology side of the company and the microbial side of the company, they're different. I think there will be a future where there's two companies. How that happens, I'm not sure. And so, but certainly, I think, the goal is really, you're a scientist, you have one life to live. You want to go after the biggest problems you can that your technology allows. One of the biggest problems, in human health, of course, is cancer. But the other one is, antibiotic resistance. In a few decades, more people will be dying from antibiotic-resistant infections than from cancer. And so that's the other biggest global health problem there is. And so if we can contribute to that, that's also pretty cool.
So Phase Genomics focus quite a bit on antibiotics resistance and also new way to do cancer diagnostic. So how is the company changing the game? Like what exactly you guys are doing that's very different?
Yeah, thanks. So it's sort of like what I was saying in the beginning. It's linked to information. So the new sort of genome sequencing technologies, what they've done is they've given us an ability to extract new information from biological samples. And so in our case, when whether we look at cancer or whether we look at microbial communities, essentially what what we are able to do is we're able to make new discoveries that push the field forward. So in the cancer space, there are a lot of diagnostics that are historically done using microscopy. They're done by looking at DNA, under a microscope to see if you've had changes, rearrangements. It's a 100-year-old way of doing this, but it's extremely important for how people analyze cancer cells and cancer genomes. And we can do it with our sequencing technology in a much more sort of high resolution way. It's much higher resolution, it's much faster, it's much simpler. And it's because, just sequencing methods have evolved to the point where you can extract genetic information so much better and so much cheaper and at such high volumes that it's overcoming a lot of the challenges that the previous last generation technologies had. So an improved way to understand what's happening in the genome is useful in the cancer space. The other side of the company, we work on, again, one of the biggest challenges in global health is the fact that we have been blessed with antibiotics, and antibiotics have been a miracle cure. They've saved untold millions of lives over the years, right? However, it's an arms race with bacteria. Bacteria, evolve resistance against antibiotics. And so now we're facing a situation where a lot of the antibiotics that we used to use all the time, most bacterial strains out in hospitals and out in the world are resistant to them.
Right.
And, think about imagine a future where, every operation could be your last one, where you can get a scratch on the, like outside somewhere, you can get a scratch or you're doing sports or something, and that gets infected and you're dead because antibiotics don't work anymore, right? And it's a sort of a nightmarish scenario, but a lot of people like the CDC, the WHO, that this antibiotic resistance problem is on everyone's mind because it is the next pandemic. And when antibiotics stop working, it's not going to be a pandemic. It's going to be every pandemic, right? Because suddenly every bacteria becomes a deadly bacteria.
Right.
And so the race is on to try to identify new antibiotic molecules or antibiotics that you can use that bacteria will not be resistant against, right? So you need a next generation of antibiotics.
Where will they come from?
I mean, they can come from all sorts of places, right? The original ones came from a fungus.
That's true.
So a lot of them actually come from fungi because fungi are good at killing bacteria. But there's all sorts of compounds that are being screened naturally and things like that. We have been extracting them from this sort of cool source. It's bacteria's sort of natural enemy. There are these viruses called bacteriophages.
Yes. Yeah.
And phages are the most numerous biological thing on Earth. They're everywhere. They outnumber everything by bazillions of times, and they kill bacteria. That's all they do. They don't kill people. They don't infect people, but they kill bacteria. And they have mechanisms for doing this, right? And they're very simple. They just have a few proteins in them. So if you can pull the proteins out of the phages that the phage uses to kill a bacteria, those proteins those are basically super powerful antibiotics. And what's nice is they don't do anything else. So it's a normal antibiotic will often, one of the problems with regular antibiotics is that they kill everything, right? So you take antibiotics after a surgery, it wipes out your microbiota, it kills all these other microbes, it kills the good bugs and the bad bugs. Yeah. But what you really want is you want something that just kills that bug that you're trying to kill and doesn't harm everybody else.
Is that right? I heard somewhere that every bacteria has a phage against them. Is that right?
Well, it has to be that it has to be the case because, if you think about how many bacteria are out there and how many phages are out there, it's this constant universe, the universal war. So there's certainly going to be a phage against every bacteria somewhere. Doesn't mean we have them. We haven't, necessarily cultured them or sequenced them or identified them, but I think that's probably true, is that they've done this study. So phages are so numerous.
Yeah.
So in the ocean, which is where most life on Earth is, in the ocean,
Right.
There's so much sort of cyanobacteria and other bacteria, and phages kill them. So about 80% of all life on Earth basically dies and is recycled every day. And it's mostly these microbes in the ocean. And so the ocean is basically phage soup, virus soup, and it's just constantly killing bacteria. And so imagine you have you have an algae or cyanobacteria that it'll absorb carbon from the air,
Yeah.
right? It'll absorb CO2. And then the phage will kill it, and then it'll die, and then it'll like sink to the bottom, and make coral or whatever. And so that so much of that is happening that phages are actually credited with altering the global carbon cycle. And so there's so many of them. So I think it's safe to say that if there's a bacteria somewhere out there, there's a phage that.
But you have to find them, right? You have to go
find them, right?
do the scuba diving. We have to go to caves. We have to go to
But
deserts.
what's cool is, so with our technology, right? What our technology does is it allows you to discover phages and which bacteria they're targeting without having to grow anything. So the problem is not that we don't have enough of them. So you can go to you can scuba dive, right, and scoop some water. But the problem is you'll bring that water back to the lab. You won't be able to grow the phage,
Right.
because the phage only grows in the bacteria,
Right.
and that bacteria only grows in the ocean. So you might have it, but you won't be able to grow it. And the reason why the sequencing technology helps you, it helps you discover these things because you can sequence their DNA even if they're not alive. And so you can get water from the ocean. We actually did this. We did one of the projects we published recently was they took microbiome samples from super deep ocean like hydrothermal vents, right, where there's who knows what kind of biology is going on down there, because there's no light, right? Their carbon source is like whatever, or their energy source is whatever is shooting out of those vents, and so you can scoop some of that material. You would never be able to grow anything that's in there because it's evolved to grow in these crazy environments. But you can sequence the DNA, right? And when you sequence the DNA from that community, you can discover the new bacteria that are there, the viruses that are there, all this other stuff. And then you can try to understand what they're doing by computational methods, right? You can see what the
So
genes
what's the
are.
big plan here? Do you plan to have your own phage bank and then help create new classes of antibiotics? Or
So what's cool is, so every phage has a protein that's called a lysin, and that's sort of like the torpedo that the phage uses to break out of the cell. So when a phage infects a bacteria, it then replicates, and then it has to break out and to infect more bacteria. And the way it breaks out is by using this protein called a lysin. And so lysins are single proteins that kill the bacteria.
Right.
And if you just take the lysin without the phage, the protein itself, you put it on some bacteria, it kills them instantly. It bursts them.
it can target it with specificity?
Yep, and it's specific to the host because that's what the phage was targeting. So for example, you can imagine you have a bacterial culture that looks like a beer. You can put a bunch of lysin protein into it, and in 10 minutes it'll look like apple juice. There are these videos that are, this is there's all these papers on it, there's classes, but there are videos on YouTube about lysins. You can watch them under a microscope slide, like a bunch of bacteria swimming around, and as soon as lysin protein enters the space, they just go pop, pop, pop like balloons.
Wow.
In front of your eyes, in seconds, you can wipe out an entire field of bacteria. So they're cool. And the other cool thing is they don't require you to grow phages. It's just a gene, and you express that gene, you make a protein, and that protein is your antibiotic. So you don't have to have phage banks, right? You can express these lysins. So I can tell you about a really cool project we're doing now. This is public information. So we're working with the Gates Foundation to, because again, we have this massive database of millions of phage genomes and lysins, and we know who their targets are. So we were approached by the Gates Foundation, say, can we use these lysins to knock down enteric methane in cows. So cows, as you know, produce lots of methane. So they eat grass, the bacteria inside their rumen chew it up, and they barf methane into the air, right? And that methane is like super strong greenhouse gas.
Right.
Like if you like, there are some estimates where if you knock down like this cow methane by like 50%, that's the same as taking every car off the road.
Wow.
In terms of greenhouse emissions.
Yeah.
It's intense because methane is so much more powerful than CO2 as a greenhouse gas. So, but the methane is created by microbes that live in their guts, in their rumen. They're called methanogens because they make methane. And so what we're doing is we're actually taking these lysin proteins that are in our database that came from rumen samples. So we have a bunch of phage genomes that kill methanogens. And if we take those samples, if we take those proteins, we can actually kill methanogen bacteria and archaea without having to have phages. And so that's a project we're working on now, is basically to use these phage proteins as antibiotics, but not against pathogens, but against bacteria that are making methane.
That's fascinating. Let's do a fun fact. A rapid fire question.
I know.
No, it's really fascinating what you're doing with bacteriophages. I always find it fascinating.
You know, bacteriophages are really interesting. There are a couple of books recently came out about the history of that space.
Yeah.
It's amazing because they've been around for over a century.
Yeah.
And they kind of show up in the public eye and then they disappear. They show up in the public eye and disappear. Right now, I think they're on the up.
Yeah.
People are really doing a lot of work in phage now.
You know, there is one very famous river in India. In the West, we call it Ganges. From thousands of years, people are going there to treat their infections.
That's where the phage therapy field started.
Yeah.
It was some Western explorer who went to the Ganges and was like, why does this happen?
Yeah.
And that's where some of the first phages were isolated from. And they didn't know what they were phages. They had no idea what it was. There was just like some particles. They thought they were proteins. They thought
it was all scientific, right? From thousands of years. Even every year now, there's like a festival. They call it Kumbh Mela, where millions of people go there.
But because it's so infected, right? Like he went there and he's like, people who have like whatever it was, like cholera or something, or diphtheria or something, and they go in there and they cures it. So like it's because there's so much of that toxin in the water, and so there's all these phages against these bacteria. So if you can pull them out, you can treat some of these diseases. But yes, it's true. I mean, that's what a lot in the Soviet days during World War II, possibly even World War I, like there was a lot of work from the Soviet side on using phages because most soldiers would die from like frontline diseases. They wouldn't even die from bullet wounds. They were dying from like from dysentery and stuff like that on the front lines. Yeah. And so they would get like corpses and isolate like what we now know as phages that target some of these pathogens, and they would like soak them on blankets and stuff like that, and it like dropped the rates of these diseases quite a lot. Right. In the army. That was one of the things that's credited to like helping the Soviets in the Battle of Stalingrad and stuff like that, just because they had this phage technology and the Germans didn't. Right. So it has a fascinating history how people have been using phages in the past century or so, but there are challenges in turning them into actual drugs. Right. Because they evolve, they change. You need a different one for different patient. That's why we're focusing specifically on these lysine proteins, because you don't have to have the whole phage, and so you just have the antibiotic property without having to deal with all the other stuff.
Can you turn them into a capsule or?
It's a protein like any other protein, so you can put them in capsules.
That's awesome. Okay, let's do some rapid fire.
All right.
If you could give your 20-year-old self one piece of advice, what would it be?
I would say stick to what you're doing, and you're gonna, what you want to happen will happen.
Love it. That's a very spiritual advice. Keep calm. It will be all right. If you had to explain your job to a 5-year-old, what would you say?
I answer emails and I tell people they're doing a great job.
Yeah. What is a biotech buzzword you never want to hear again?
Ooh. Biotech buzzword. Oh geez. I'll have to pass. I mean,
That's okay.
something about AI, but I'm sure.
Yeah? Oh, something about AI. Oh boy, I'm going to cut it out, If you could fix one major issue in the healthcare system overnight, what would it be?
I think the cost of clinical trials is probably the biggest bottleneck to all therapeutic development.
Cost because
It's
of
so
failure
expensive.
or
Everything.
recruitment?
There's a lot of regulation. Regulation is very, very heavy. It makes it so expensive to do any kind of therapeutic or diagnostic development that a lot of times you have really good things that just never make it to the market.
Very true. If you had to bet your entire career on one biotech trend, what would it be?
Ooh. Oh man, these are hard.
I thought they were fun questions, no?
They are fun, but it makes me now I feel like I can get it wrong. I can say something. This is an opportunity to say something stupid. I think getting more genomics information into the medical world is something that it is a trend. I mean, it is happening, but I
for
think
diagnosis or
for everything, for diagnosis, for therapeutic development, for patient stratification, for all sorts of things. I think just maybe let me summarize it this way: like better information makes everything better.
100%. 100%. I sometimes think people wear these wearable devices. If we can connect this with our genomic data, our immune profile, our all other proper blood function, then what can we do with that, right? We can make better decision, even what to eat.
And doctors too, right? Like doctors need to know.
Totally. Is entrepreneurship a skill or a mindset?
Entrepreneurship is a mental disorder. This is what I often say, is that I'm a CEO, I'm a founder, and I'm an entrepreneur. A founder is just a historical statement. I founded a company, I'm a founder. CEO is a job.
Yeah.
I have certain tasks I do for money, and entrepreneur is a mental disorder that makes you
have to be crazy.
You have to do this and you can't stop.
Yeah. I think you have to be crazy to start something from scratch and then stick around for a decade or so. No, totally. What is your ultimate vision for how genomics will transform human life?
Oh man, I think it'll transform it in many ways. I mean, I think the most kind of straight answer is we'll know more about our bodies and about our health, and so we'll be better equipped to deal with diseases and things like that. But there is lots of other. There's agricultural improvements, right? There's environmental improvements. There's, I think it'll impact almost everything we do, right? Our health, our food, our, transportation with like
Biofuel
almost
or
everything.
something.
Yeah. We can think
But
of.
what's the promised land? Like if you could live to see 200 years in the future, like how do you see is that like promised land? Let's say we leverage genomics at its like full potential.
Sure. Well, I think at least within the health sphere is when you have holistic understanding of everything about you, then maybe you eliminate all disease, right? Or you eliminate most chronic diseases that we don't understand. If you were able to prevent problems before they happen, right? Like a lot of folks are trying right now to do really sophisticated testing to try to predict like what kind of foods you should eat and things like that. But there is evidence that like not everything needs to be cured after there's a disease, right? If you knew more about yourself, you might be able to prevent things that right now seem to be like impossible. Can we leverage the information that we get from genomics, not just to be better at curing problems, but better at sort of preempting them and defending them, so that defending against them before they become a big problem.
I mean, that's what healthcare is supposed to be, right? Currently, healthcare is all about fixing human bodies.
Sure.
It's not about preventing, like basically diagnosing before it happens. That's what, if you go 1,000 years ago and see our old civilizations, in like Ayurveda, it was never about fixing human body. It was always about preventing it even before it happens.
Yeah. The problem is it's hard to measure when something doesn't happen.
Right?
Like
that's
you're
true.
like, so that's the issue. And of course, obviously we're incentivized to make things we can sell.
That's true.
And prevention is hard to sell. A cure is much easier to sell than a prevention.
Like diagnosis business is harder.
But I think it's also all sorts of other technologies, right? Like food security, right? Fighting off pandemics in the developing world and here, right? Climate change, affecting climate change, like what I was talking about, the enteric methane. I think it's, improving food supply. Remember, like in the 80s, there were all these books and papers about how we were going to starve by the year 2000. There's too many people, not enough food. And the Green Revolution really fixed that. Like, we don't think about that anymore.
Yeah.
That was a biotechnology kind of breakthrough. It was a lot of plant breeding and things like that. Can we make the world a better place for everyone? Here, in other parts of the world, on the food side, on the environment side, fix climate change, improve our health. I think all these things touch biology, and biology is just information. And so better information arms us to improve, everything that we live in, and our own biological processes, and things like that.
What are your thoughts about aging? Do you think that's a disease?
Is aging a disease?
Yeah.
I mean, I think that aging is a biological process. It is not like the slow breakdown. It's not like car gets rusty over time and things just gonna fall off. I think there are specific biological features. Like there's something about us that ages. Like it doesn't happen by accident. Like our bodies age for a reason, and so it's a process. It probably can be slowed, maybe even stopped. Who knows? I mean, it's a big question, but it's clear, I think, from the science is that it's not just stuff breaking down over time.
Yeah.
There's some biological stuff that's going on that makes you age, and maybe there is an evolutionary benefit to aging.
Right.
And maybe we evolved aging for a reason. I don't know.
Comes with wisdom. I love that.
Yeah, maybe.
Would you like to live 300 years? No?
It depends. We might. Other people might not, right? Like, other people might be like,
No, I like this sense of urgency, right? We all have limited time. And arguably that's what makes us ambitious. Because we know there is a clock and we have limited time, so we better do something.
Yeah. I mean, that's definitely one of the features. I don't know if that's why we age, but that is for sure a feature of our lives. And I think it sort of gets into the question of what are we trying to do as a society in general? Like, what do we want? Right? I think biology is such an inherent part of everything that we live with, that we are biology. Right? So obviously we want to improve that as much as possible.
Right.
It'll help all sorts of things. And so all these new technologies are, giving us more tools,
right?
And we're understanding how our bodies work better, and so that allows us to create new ways to deal with problems, new ways to predict and prevent problems, new ways to create just like useful things, like there's flowers that glow and stuff like that just for fun. There's
Makes
also
sense.
that.
Yeah. No, love it. I think so much genomics wisdom. I really appreciate it, Ivan. It was really fun.
Thank you. Thanks for having me.